Okay, let's take a look at these NMR practice problems. So in this first question, they're asking you to select the molecule that gives the proper splitting pattern. What we see are here they're giving us the integrals, and then on the right-hand side of that, they're giving you the splitting pattern. And we're just trying to see that which of these molecules would line up with that. So generally speaking, so for example, if you were working at this systematically and just going through each molecule, you would start with the first one, and you would say, you'd look at all the different protons in it, and you would see if they line up. So what would I expect from this molecule? Let me label these protons. Yeah, for the orange versus whatever this color is, red brick or something, versus the pink, and versus this green. I'm going to have to erase this because I'm going to run out of room, but what would I expect to see? So for the first one, for the CH3 group, I'm going to expect to see something that is 3 hydrogens, but that is also a triplet. And then for the one that's kind of a red brick color, I would expect to see 2 hydrogens, and that would be a quartet. And then for the other methyl group, the one that's in red, I would expect to see, so this is one thing I don't think I talked about in the videos, but I would expect to see 3 hydrogens. You won't see any coupling with the hydroxyl group typically. It's not always true, but usually you just don't see it. So just assume that you're not going to see it, but don't be totally shocked if it were to show up in an NMR. I mean, it probably won't show up in the quiz that way, but just for the second course or whatever. But yeah, so we've got 3 hydrogens there, and they're just going to be a singlet because they don't couple, as far as we can see at least, to that hydroxyl group. And then lastly, in this green proton here, again, this one doesn't couple with anything, so what you end up just seeing is 1 hydrogen. And that usually comes out as a singlet, and it comes out as kind of a broad singlet. So usually when you're looking at the NMR, you'll see peaks that look like really crisp. They come to peaks like that. The tops are like, like the function isn't continuous through that top part, to use some terminology from calculus. But for the alcohol, what you'll end up seeing is something that looks more like this. So the signal is kind of like spread out and looks more like an anthill or something. But it's not always like that because nothing's ever straightforward. But yeah, so this is what you would expect to see, and then we come back up here and we see like that doesn't really match up what we're seeing here. Like, yeah, we've got, maybe you can match up the singlet, but we don't have any triplets or quartets here. Like, we got a doublet, no net, doublet. Here I have a triplet, I got a quartet, I got another singlet. So we can tell that that's not the right one. So that's one way to go about doing this question. And you could go through each one of these answers and sort of converge on that, on the correct answer. And I think that's fine. I think we probably give you enough time in the quiz to go about that route. For me, because I have experience with this, I just kind of like, some things just stand out to me as important. This doublet, like the 6 protons that are a doublet, usually if you have like a bunch of hydrogens, so if you integrate, so if you're looking at your NMR and I've got whatever, some sort of peak out here, be it doublet, triplet, or whatever, but the integral of that is 3 or 6 or 9 or something like that, some multiple of 3. It usually implies that I have a methyl group, or if it's a 6, if it integrates to 6, then I'd have like 2 methyl groups, maybe like an isopropyl group. Or if it were 9, then I would expect to maybe see something like a tert-butyl group on there. But multiples of 3 usually imply symmetry. If I just have 3, it implies just 1 methyl group. If I have more than 3, like 6 or 9, that implies multiple methyl groups, but some sort of symmetry in the molecule that makes them not unique. So that stands out to me as like, a good indicator of what's going on there. So I'm expecting, when I look at this, the thing that stands, that not all the evidence is equally important. The 6 protons that are a doublet, to me that implies 2 methyl groups. So I'm looking for 2 methyl groups. When I look through these, I'm looking for 2 methyl groups that have some sort of symmetry in them. And what I see is the only thing that has that are these 2 molecules down here. Okay? And so if we look at this, and this is probably the most common way to see that signal of 6 protons show up, is you have an isopropyl group. So remember I said that if it's an integral, if it's a multiple of 3, so 3, 6, or 9, it's most likely a CH3, an isopropyl, or a terp butyl group. And that's not always true, right? There are different ways that you could get 2 equivalent methyl groups in a molecule. For example, if you had something like this, that would also give you a multiple of 2. So this would also integrate to 6 or something. So you can imagine, or like maybe something like this. There's lots of different ways to get 2 methyl groups that would show up as under one signal inside of a molecule. I'm just saying that one of the more common ways is to have an isopropyl group, and it seems to be true here in this example as well. So both of these satisfy that. The isopropyl group's symmetric. And then I would look just like next door and say, well, what am I going to expect to see for that proton? Because that proton is going to be pretty telling. So in the one on the right, in the one on the left, where I marked it in green, I'm going to expect to see a septet. Sometimes if it's, you'll hear these, like if it gets to be, the splitting starts to get really intense, like nonet or decad or whatever, like these really intense, or like a septet of doublets or something, you'll just see it written as multiplet. There's none of that up here, so we're just assuming that they're going to tell us exactly. Yeah, I look at this one, and what do I see? I see that this proton is going to be split not only by these two to make it a septet, but there's also these other 2 protons there. So I'd expect to see maybe like a septet of triplets or a triplet of septets, or if it's just following the simple n+1, I would get 3, 6, 7, 8 plus 1, so I'd expect to see a nonet. And I see up here I have a nonet, whereas for this, there's a septet. There is no septet up here, right? So that allows me to very quickly converge on this as my answer. What you would want to do is probably go through each of these and make sure that you can convince yourself that, for example, there's no way that this could line up, with this set of signals here. And I encourage you to do that. But yeah, for me, I sort of hone in on what the important details are, just because I have some experience, and I'm trying to, I don't know, I guess inform you of like what those are. I guess the only other thing I'd like to point out about this problem, because I don't think I went over this in the other videos, is actually, let me save it for another one. I want to talk about alcohols a little bit, though, at some point. Actually, no, this is a good one to do, because that is the actual answer. So if I look at the answer for this, it's answer 5. And a reasonable question is like, why? Let me clean this up a little bit, because I can't see what's going on there. Yeah, okay. A reasonable question is like, I've got a proton on the oxygen. Why is this proton not show up? Why does this proton not show up as a triplet? Right, because it's next to these 2 protons, and that's what our n+1 rule dictates. But yeah, so why is it not a triplet, right? Like what we actually end up seeing is a singlet. So this is really common for hydroxyl groups. It just shows up as a singlet. You'll also notice that in the spectrum, like these 2 protons here, if they were coupled with this, the proton I've highlighted in red on the alcohol, you would expect to see a doublet for those. I'm sorry, we've also got a hydrogen over here. So you'd expect to see a doublet of doublets or a quartet or something. But in actuality, we just see a doublet for those. So there's clearly, like, at least practically we don't see it. There's no coupling between those 2 protons. And that's just really common for alcohols. So alcohols, we don't always see coupling, or don't always, I'll just say call it splitting. We don't always see splitting. And so regardless of what the alcohol is next to, it'll usually appear as a singlet. And moreover, it'll appear as a broad singlet. So a lot of times when you look at the NMR spectra, they'll be crisp at the top. They'll sort of have this, the function won't be continuous. It'll sort of break really. The slope will just change really rapidly. For alcohols, you end up seeing something that looks more like that. So it looks more like, just like a normal, distribution with a curvy little top on it. So alcohols are just kind of weird. I guess that's the main thing here. And you don't usually see them splitting other protons, although you do sometimes see it. It just depends on it honestly, it depends on the solvent that you're running the NMR in and some other factors, but we don't need to get into that. But yeah, so that's how you would answer that one. And this next one was nice because we actually get a spectrum to look at. So in this one, how you start these is up to you. You could start by looking at the hydrogen deficiency index. There's a bunch of different ways we can calculate that. If you want to use the formula or you want to just kind of eyeball it like I like to do. The HCI for that comes out to be 5. That doesn't help us that much, though, because all of the molecules that are choices have an HDI of 5. That's more of utility if we have no choices or if some of the choices had different hydrogen deficiency indexes. So it doesn't help us narrow things down. The next thing I would look at would be the integrations. Those can be pretty telling. The integrations in the number of sets of signals, I guess, is what I would say. So for this one, we've got 1, 2, 3 unique hydrogens in the molecule. If we come down and look at these molecules down here, we can disqualify some of them at least. Let's see, which ones can we disqualify from that? So this one would have, it has 1, 2, 3. Sometimes it doesn't show up, but let's call it 4. And then it looks like the aromatic portion of this molecule, you would have unique signals there, but it doesn't look like it's being resolved. So we're going to call that 5, even though they're not perfectly equivalent. I'm just looking at the spectrum and noting that this is the aromatic region out here because it's above 7, and it's appearing as one signal. So even though they're not equivalent, the resolving power of the NMR is not enough to actually resolve out all those different signals there. So those are all just being counted the same. But yeah, so we've got 5 in this one, 5 different protons, and so we would expect, 5 different signals here. So that's one thing that starts to clue us in on like what's probably going to be the right answer or not the right answer. Another thing you can look at is, the next thing I might consider is the integrations here. So remember what I said, if you've got 3 hydrogens, that usually indicates a CH3, especially when it's appearing down here. When it's starting to get into that 1 to 2 region, that's really where CH3 shows up. And so we're expecting a methyl group on one of these. And you immediately, that's maybe the first thing that's really helpful here is this one, there are no integrals that would be 3. Same thing is true for this one. Right, I have nothing that's worth just 3 protons. My handwriting's pretty bad there. I have nothing that would appear as just 3 protons here. At least down here I have a methyl group and I have a methylene, so maybe that one's right. This one I have several methyl groups, so it could be those. I've got a methyl group here, but at least this one allows me to get rid of this, so I know it's not that one, and I know it's not this one. But then I can keep going with that logic and think about some of these other ones, like this methyl group corresponds to 3 protons. So if I have another methyl group in a molecule, then I should see another signal somewhere in here that also corresponds to 3 protons, but I don't. So that immediately disqualifies this one because I can only account for one of those methyl groups in this spectrum up here. I only have one signal that actually has 3 protons underneath it, so I quickly disqualify that answer as well. And then I'm just left with 2 choices here. So let's see. So the methyl groups I think we've sort of exhausted in terms of their utility. Now we have to go and we can look at the splitting patterns for these. So if I look at, I've got a methylene, let's see. Yeah, the splitting pattern will tell us here. So here I've got a methylene, and here I've got a methylene. So I should have an integral of 2, which I see up here. So, if I'm trying to figure out what this could be, it's like, well, okay, that could correspond to this or this. But we notice that our splitting pattern here, it's just a singlet. So if that's a singlet, that immediately disqualifies this one because this one is next to 3 protons, and this would give us a quartet for those. So that is clearly not going to be that proton, which leaves us with 4 to make it as our correct one. Yeah, what does the aromatic region tell us? I mean, it just tells us that we have 5 protons out there, and we see that that's true. I would expect to be able to see the splitting on these, but we don't actually see it, so you just assume that the NMR is not powerful enough to resolve that splitting. That's not uncommon. But yep, that's how you get to that answer number 4 being correct on that one. This next one I kind of like, because this is more, this is kind of like what things are like in the lab, where you're like running a reaction, and then you're using, usually when you're using, what you're doing is you're using NMR to confirm that you actually got the molecule that you were hoping to make. So before you even get down to the NMR, you have some idea of what you were trying to make. Just to point out, like here, we're doing the halohydrogen reaction. We're going from that alkene to get to the halohydrogen. We don't see any, even though you've got beta branching right here, we don't see any rearrangement, because remember, for the halohydrogen reaction, it's much less common to see rearrangement because you're not actually forming a carbocation, you're forming a bromonium intermediate, so your intermediate there is this. So if you're forming a carbocation, you might expect to see rearrangement. That's something you got to watch out for when you have beta branching. But here, we don't see it because we don't actually form a carbocation, or at least that's the assumption we can make. But yeah, we come down and we look at our NMRs. And we see what I've kind of been talking about before, which is that if I've got an isopropyl group on the molecule, typically it just shows up as 6 hydrogens somewhere far up field. And we see that here. It doesn't actually help us solve this spectra, but we see signals that are consistent with an isopropyl group. We see the 6 hydrogens, and then we see some splitting. We're just going to call that a multiplet because I can't tell what's going on there. But we see a multiplet that's nearby that corresponds to this proton. Right, so that's that showing up. And so that doesn't help us, but it's consistent across the spectra. At least we know that the product we got isn't insane. So then we just go to some other protons, though. The only other 2 protons we have are these. And so we're expecting one set of signals corresponding to those 2 methyl groups, another set of signals corresponding to the green hydrogen, and then one last set of signals corresponding to this new methylene group that integrates to 2. And so when we look across our different spectra down here, and we look at these different ones, the only one that could possibly make sense for this then is this one, because it's the only one that has an integral of 2 for it. And also we can look at the splitting pattern here. We know this should be a singlet. We see we have some singlets that are pretty far downfield for these, which is probably what we would expect, because this is sandwiched between a bromine and a carbonyl group. So both those electronegative groups are going to push things farther downfield. I don't know that you would expect it to push it all the way above 12. That's usually like where we see like aldehydes or maybe carboxylic acids or something. But yeah, regardless of what the shifts are, we also see that we have extra signals, right? If we look up here, we're expecting to see 3 signals with whatever splitting. The only NMR that corresponds to this is this one. These all have extra signals in them that we can account for based on what we're expecting in our product. And so the correct answer for this one is A. Okay, so here we're just trying to figure out how many unique hydrogens we have. And so you can do this exhaustively using that substitution method I teach in a different video. Eventually you get to the point where you can kind of build up an intuition for this. This is what we've been doing all semester when we talk about radioselectivity. Really what you're saying when you're talking about radioselectivity is you're kind of indirectly, even though you don't realize it, you're talking about how many unique positions there are in the molecule. So that's what that substitution method is really doing. But yeah, so how many unique sets of hydrogens do we have here? So for isopropyl groups, usually these methyl groups are interchangeable, and this is no exception. If you don't trust me on that, you can go ahead and do the substitution method. This proton is the only CH group in the whole molecule, so there's nothing that you could even question what it would be equivalent to. Same thing for the methylene group. Those 2 hydrogens are the same. If they were near a chiral center or something, we might have to worry about it, but they're not. And then the last situation here is just these 2. These are maybe the 2 hardest ones to see. So yeah, that teal and purple proton are actually different. And there's a bunch of different ways you could rationalize this. The main reason that's true is because there's no free rotation around this double bond. So, these methyl groups get exchanged sort of because you can spin across that double bond, the single bond, and that's why they're not equivalent. But the double bond over here, right, we don't have free rotation. And so that's why those two hydrogens are not the same. And if you want to test that, so you can just say in general, this is generally true for a molecule with this sort of stereochemical configuration where you've got one side of the alkene that's different. So there's one more hydrogen here. But where you've got one side of the molecule is substituted. So if you have an alkene that just has one substitution on it, or it could have more than that, but that starts to create a situation where the protons are no longer equivalent. And so one way to think about it without having to get exhausted, do it exhaustively, is like if I'm standing on this proton, that what makes the protons unique is whatever you want to, like to be really detailed, is like if they're in the same sort of magnetic field or whatever, right? But really what we're saying is like if I'm dropped in from a helicopter onto that proton and I'm dropped into a helicopter on this proton, can I tell the difference? And the answer is yes, because if I'm here, right, when I look around, I'm going to see that R group. And when I look around, when I'm on this molecule, what am I going to see? I'm going to see a hydrogen. So I'm seeing different environments there. If I were to compare this to a non-substituted one, or something, let's say, where the substitution is the same, so we've sort of restored symmetry here. Now I can't tell, right? If I drop in here, when I look across, I see a methyl group. If I drop in here, what do I see? I see a methyl group. So for all practical considerations, like these are identical and they show up identically in the NMR. If I want to use the substitution method, though, because I don't understand what the hell these little guys that I'm drawing dropping in from helicopters are, we can do that too. So if I were to take this and subject it to two different conditions where I were to replace one of those hydrogens with deuterium, you could use a bromine or a methyl group or whatever you want to do this reaction. It doesn't matter. I see that I get two different products. So in this one, this R group is trans to the deuterium, whereas here the R group is cis to the deuterium. So clearly those two positions are different if doing a substitution at those leads to two different products. That means that those two hydrogens are not equivalent, right? If they were equivalent, then I wouldn't expect to get two different products out of it. So anyway, so now I go through and I just count up how many different unique hydrogens I have. I have 1 for the orange ones, 2, 3, 4, 5, 6 sets of hydrogens, and so the correct answer here is 2. 6 sets of unique hydrogens. So you can be extremely exhaustive about this by doing the substitution for each proton. Hopefully you can get to a point where you don't have to do that for every single one and it becomes a little easier. And I think I talk about that in the video I made for this. Okay, so for this one, I think on the exam, we give you, we label the different unique hydrogens for you already. Regardless, you guys know how to do that. We just did that. Let me go ahead and label them for you, though. So these are unique. This is a tert butyl group. If we were going to look at the NMR for this, we'd expect to see one peak for that. That's a singlet that integrates to 9. Kind of getting back to that first thing that I talked about, which is CH3 groups show up as multiples of 3 in the integration. And if there's some symmetry, they show up together. We don't have to worry about that for this problem, though. So we've got 4 different unique hydrogens here, and we're trying to figure out what we're going to see for each of these. Oops. So let's start with the red hydrogens. What are we going to expect to see for those? So we're going to expect to see an integral of 9, so there's going to be 9 hydrogens there. And then what's going to be the splitting pattern there? We're just going to expect, let's see, how many protons are on this carbon here. Hopefully you're not drawing any, because that already has 4 bonds to it. If I can get some better precision with my eraser. Yeah, so I got 9 hydrogens there. They're not split at all, so I'm just going to expect a singlet for that. Sorry, I'm looking at that. I don't see that in the answer choices. It's weirding me out. I'm making sure I'm right, that I didn't draw the molecule wrong. Yeah, I think I'm going to add that in here. So there's broad singlet, but that's not going to be broad. This will be a normal singlet. I don't know. Maybe it's broad. I guess if I was on an exam and I was forced to pick one, I'd say, this is probably the best option for that one. So let's just go with that. But yeah, so we've got a singlet there. What are we going to expect for the other ones? Yeah, so for the green one, this is something we didn't talk about in the other videos. I'm glad it's coming up here. Every unique, so usually what we talk about is this n+1 rule, where it's like I'm on a carbon and I've got a hydrogen. I look at the carbons next door. Right, the carbons on the same hydrogen and the hydrogens on the same carbon don't split it. So what splits in this molecule I just made up, propane, what's going to split these hydrogens? They're going to be split together, right? So I'm going to expect to see this n+1 rule situation where I get 3 plus 3 is 6 plus 1 is 7. So I'll expect a septet. But if those protons were different in some way, so if they were diastereotopic, so if instead of having a methyl group here, I had a chiral center of some kind. I don't know what the chiral center would be, but now I break the symmetry of that. And now these protons, now these protons are different from one another. Oh my goodness. Damn Apple Pencil. Erase the other color. I'm just going to keep changing these colors back and forth and erasing them. There we go. All right, so now these two protons are different, so we expect these actually will split each other, even though they're on the same carbon atom, right? So diastereotopic, let me write this out cleanly. So what I'm saying here is that diastereotopic protons on the same carbon can split each other. So when you're on an sp3 hybridized carbon, like this example I've drawn here, usually that splitting is hard to see in the NMR unless you've got a really, really powerful, well-resolved NMR, or really powerful NMR. You spend a lot of time resolving the image on it. So it doesn't always show up in that. It may or may not. But for alkenes, which is what we're looking at here, you almost always see some sort of splitting for it. So this proton will be split by this proton. And if you go in the book, there's a lot written about that. Alkenes they're pretty common functional groups, and the NMR splitting patterns for them are pretty well understood. So, but yeah, what do we expect to see then? So it's going to be split by that proton, and it's also going to be split by its next door neighbor proton. So that's still, that carbon over there is still going to, the hydrogens on it are also going to split it. So you're going to expect a doublet of doublets, right? So it's going to be split by this, which is going to give you a doublet, and then it's going to be split by that, which is going to give you a doublet. So you get a doublet of doublets. Or it could also be a quartet. So I don't know. Hopefully on the actual quiz it's not ambiguous, but if I had to say definitively what I would get, like if I had to make a guess here, I'd say doublet of doublet. I guess what I'd say is if this was on the quiz, either one of these answers would be fine. Because we don't know if it's going to be a quartet or a doublet. It's how it's going to show up necessarily, right? But I'm just going to go with doublet of doublets because I think that's what they wanted. I actually have a key right here for it. Yeah, doublet of doublets, what they wanted. What did they want for the other one? Oops, not what I meant to do. Yeah, 9 protons is a singlet, okay. Let's see, and the purple one, purple proton's going to be in the same situation. It's going to be split by the green hydrogen, but it's also going to be split by its next door neighbor, that blue hydrogen. So we're going to get a doublet. We're going to need a doublet of doublets for that as well. Or a quartet. Both of those answers would be correct in this situation. And then finally, for the blue hydrogen, that's going to be split twice. And again, for the same reason, it's going to be split once by this, but it's going to be split again by that. So, for this one, well, I guess in this case, you'd see a doublet of doublets, because it's going to be split once by this or once by that. Is that right? Yeah, doublet of doublets. Or you'd see a triplet. Since I looked at the answer key, I'm going to call it a doublet of doublets. But again, I don't think any of the questions on the quiz are this ambiguous. But, again, you might expect to see a triplet, or you'd expect to see a doublet of doublets. And we can check what the question writer was interested in. Yeah, they thought doublet of doublets for everything. And so that's the answer to that. Yeah. The main thing here is that when you have diastereotopic protons on the same carbon, especially in an alkene like we're seeing here, they split. They like contribute. They'll split each other, and they also affect their protons that are nearby, like that blue proton. They each have an effect on that one as well. And sometimes you can see it, and sometimes you can't. So maximally, what you'll see is a doublet of doublets. But you might end up just seeing. You might end up just seeing a doublet or something like that. It kind of just depends on the NMR. It's kind of like if you look through a microscope, right? It's just what you actually see is sort of dependent on the power of the microscope. Like if I take a leaf and I put it under a microscope, depending on the power of the microscope, I might just see a bigger image of the leaf. I might be able to start to see some individual cells in the leaf. If I have a really powerful microscope, I might be able to see even finer structure in the cells than I would otherwise. In the NMR, it's the same thing. It just depends on how powerful the NMR is. So maybe I end up seeing a singlet, even though if I had a more powerful NMR, I'd see like a doublet of doublets. And it just kind of depends on what sort of the magnifying power, in a sense, is of the NMR is. So it's sort of it's a good analogy, I think, to the microscope or telescope or something, right? It depends on the power of it as far as what you actually see. So if you zoom out really far, especially in a leaf or something, you don't see any fine structure. It looks like one continuous thing. And that's sort of what we see when we end up just seeing like a singlet or something where we might predict a triplet or a doublet of doublets or whatever. Okay, same kind of question. This one circles back to that alcohol thing we had talked about. So for alcohols, we generally expect to see, and I don't think we have any alcohols on the quiz, that at least not in this particular format. But generally what you expect to see is a broad singlet for that. So we're going to expect to see that. So we'll go ahead and mark that one just right out of the door. So we expect to see a broad singlet usually for that. That's not always the case, so it depends kind of on the solvent and stuff. But for this question, let's just assume that. On the quiz, we won't be that ambiguous, but let's see. So we can look at our other protons here. What do we have here? So we've got 1, 2, 3, 4 different signals. We've already accounted for the alcohol. What are we going to expect to see for these other ones? This one's a little bit more straightforward than the other one. So here we have that isopropyl group, which we've seen before. So we're going to expect to get, for 6 hydrogens there, we're going to expect to see it split by this proton here. So what are we going to get? We got 6 hydrogens, but it's going to be a doublet. And so we come up here and we see that that's a doublet. That matches that perfectly. If we look at this hydrogen, what are we going to expect to see for that? Well, we're going to get split by this. So we're going to be like maybe a septet, and then it's going to get split again. So you might expect to see like a septet of triplets or a triplet of septets. A lot of times what that ends up looking like, so like a triplet of septets, right? So a triplet looks like this. And then imagine that I split each of those into 7. It ends up looking something like this. Or if you had a septet of triplets, like a septet looks something like this. And then imagine that I split each of those into a triplet. It starts to just look kind of like a garbled mess. And if you have a really good NMR and you zoom in and you're really careful about how you do things, you can actually resolve that. But a lot of times it just ends up looking like a little bit of a mess, and we just call that a multiplet, right? So that's when it's like you can see there's a whole lot of peaks there, but you can't quite count them, and you can't see if it's a septet or a septet of triplets or what. So we just call that a multiplet. So multiplet's our best answer for this one. And then finally we just have these green protons here. We're trying to figure out what those are. So remember, we're not expecting those to couple with the hydroxyl group. That's unusual for us, at least. But it will couple with that proton. So we're going to expect to see a doublet. Yeah, we'll expect to see a doublet for that. So we'd answer this twice. And I think that's pretty much it. And we can check our little answer key over here. So yeah, so we get a broad singlet. That's the alcohol group. We have two protons that are a doublet. Let me outline these. That's going to be the doublet. That's going to be the broad singlet. This is going to be the doublet we talked about. And then finally, just this weird multiplet is going to count for that proton. So I think that's all the problems, yeah. So yeah, so these, I think for the practice problems, maybe we just didn't get far enough along to kind of like put them in the perfect quiz format. But the questions on the exam should look very similar to this. I don't expect it to cause. Hopefully, it doesn't cause a lot of confusion. Presumably, these are labeled as well on the quiz. Yeah, but that's how you work these. So the typical, really, like when you get kind of on since I guess this is the last thing I'll ever talk to you about with NMR. But really, when you get on in the course, right? This is really what you want to be doing. You want to be looking at squiggly lines, to use the term that another professor used today, in being able to kind of translate those into something meaningful, like trying to assimilate the data in those squiggly lines and trying to say, what does that molecule look like? And for those of you going on to be medical doctors, there's a lot of that happens as a medical doctor. Like if you go into a patient's room, you see a lot of like weird squiggly lines, and those are all telling you something different about that person's vital signs. And you actually do it. You actually will use NMR if you're interpreting like an MRI. I don't think you ever have to interpret the signals, but MRI is based on NMR technology. So this is getting closer, I think, to something that's a little more relevant, I think, to your careers. If not, it's obviously not exact, but the logical progression, I think, from going from a weird analog signal or something that you're trying to monitor to or squiggly signal looking thing that you're trying to monitor to something that's actually meaningful. That part is certainly something you'll do for your rest of your career. So anyway, all right.